different in that when we talk about carbohydrates or proteins, we talk about a distinct chemical class, whereas fats really has quite a variety in terms of the chemical structure of things that are called fats. And it's more their sort of physical properties, namely the fact that they bind water very poorly or not at all. So we say that fats are hydrophobic, kind of meaning that they fear water, which is not true. It's not that they don't like water. It's that water doesn't like them. It doesn't associate readily with them, only when forced to. All right, so the fats have to stay by themselves most of the time. That's why fats glom together. That's why oil forms a big bubble at the top of the water in the pan. Okay, so we're going to look a little bit at the varieties of fats and then talk a little bit about their metabolism. So most of the fats that we get in our diet come in this form. What if I kind of stick it here? Okay. So these are called fatty acids because the acid part, this OH, has a structure such that this H can be released to make H+. And that's what acid is. Acids is protons, charged hydrogen, H+. And so this is the acid end of the molecule, and this will bind water. However, if you look at this part, the key feature of it is that there are no oxygens in there. And that means that the electrons in all of these atoms are distributed very evenly over this whole chemical structure. And so it develops no strong charge. There's no place along here that's strongly positive or negative. And therefore, it has no affinity for other things that are positive or negative. It's a very neutral compound. And this is kind of the fatty part of this molecule. So these exist in long chains like this, and they come in a variety of forms. The principal ones you hear about in your diet are saturated fats and unsaturated fats. And I'll show you, actually the word fat is a little more complex than fatty acid, and we'll come to that in a minute. So saturated, filled with hydrogens. So every possible hydrogen binding site in this chain has a hydrogen atom. However, you do get natural fatty acids in which that is not the case. So this carbon here, instead of having two hydrogens bound to it, has a double bond to its neighboring carbon. And this one has one hydrogen and a double bond to a neighboring carbon. So we say that this is unsaturated. It's really missing two hydrogens that could potentially be there. Now this comes up in the diet a lot because, all right, which are the good ones and which are the bad ones? Is this the good one or the bad one?
Good one.
That's the good one, all right. And they're more abundant in plants. And this is the bad one. Now, to grossly oversimplify it, it's largely because this kink in the molecule makes it more difficult for two fatty acids to bind to each other because they can't line up evenly. So they are less likely to clump. They're less likely to form a solid. So oils will have a lot of these unsaturated fatty acids, whereas solid kinds of fats like lard or butter will be high in the saturated fatty acids. And you can see this if you look at a molecular model for that. So here this is just another way of writing that carbon chain. So this is an atomic structure where the black is the carbons and the white are the hydrogens. And this is this acid group at the end. So if it's fully saturated, it's a fully straight chain. But if it's unsaturated, missing a couple of hydrogens, you get a kink in the chain. All right. And it's really as straightforward as that. So why do plants have unsaturated fatty acids and animals don't? It's because fundamentally, the hotter it is, the more the butter's going to melt, right? All right. So things are going to be liquid more likely, less clumped at higher temperatures, like 98.6 degrees, which a lot of bodies run at. But if you are a plant, if you are a redwood going through a Santa Cruz summer, all right, you don't want all of your fatty acids lined up forming clumps because you need fluid membranes. And so the way this is evolved is they have high levels of these unsaturated fatty acids, which tend to stick to each other much less readily, and they're more fluid. Okay, and whether that translates directly into whether you get these accumulations of fat in your arteries, etc., it's probably not nearly that simple, but that is the kind of core underlying reason why saturated fats are less good for you. They just tend to be less soluble. Now, there's another type of fatty acid. They do occur in nature, but they're very low abundance, in the order of 1%, in which you have a double bond, but what we looked at before was the two hydrogens were on the same side as cis. On the other hand, if the two hydrogens are across from each other, across is trans. And this is the basis of trans fats. So you may have heard about trans fats in the diet. In fact, in the last couple of weeks, they've been in the paper because there's a new UN resolution of some sorts to eliminate trans fats worldwide. So when you get this trans fat, you get something that's straight, but it's just got a little kink in it, look like this. All right. And exactly why trans fats are not good in the diet is not really well understood. And the reason for the banning of trans fats is basically epidemiology. And just that there's very strong evidence to show that a high consumption of trans fats is highly correlated with a high rate of cardiovascular disease. And the banning of trans fats has been correlated with a drop in cardiovascular disease, although that is due to other factors also. But there's a very solid consensus that this is bad for you. It probably has something to do with the fact that we don't have the enzymes to deal with them because we rarely encounter them in a natural diet. Where these come about is that the cheapest, easiest way to get a lot of fat as a food source is from plant seeds. All right, but if you want to make margarine, or the major place you got this is from Crisco. Everybody in this room knows Crisco, right?
Yes.
Well, people still bake. Crisco no longer has trans fats. What the food processing people would do is they would take these unsaturated fats and hydrogenate them. They would add protons, but they don't do it with enzymes, they're doing it chemically. And so when some will come out with the extra hydrogens on the right side, but others as a byproduct, you'll get these trans fats. And so they came about from partially hydrogenated corn oil. You would often see this in the past. And it is possible, and now trans fats have been banned in the US for, well, the ban actually took place about 2 years ago, and it has 3 years to be fully implemented. But for longer than that, there have been very few food products that have trans fats in it.
So why were trans fats used in the first place?
But, because it was cheap, again. It's an easy, cheap way to get things like margarine and baking. What's Crisco? What is it? Shortening. No, why it's called shortening, I do not know. Does anybody know why shortening is called shortening? Okay, other questions about the trans fat?
Is there evidence that if you cook at a high heat with olive oil or other low temperature fats, that you can make trans fats?
Not really, not to any significant amount, because what you really need to do is kind of add molecular hydrogen, or if you do it in an expensive way, some particular chemical hydrogen donor. So that's not a problem. Other bad things do occur, however.
Such as?
high heat can cause things like starches, etc., to form a compound called acrylamide, which is a neurotoxin.
But that's a starch, that's not a fat.
What's that?
That's a starch.
Yeah,
that's not a fat.
so you can get certain kinds of nitrate thing. Anyway, you're at really high heat, you start doing chemistry, and instead of biology. And so you just get a low level of toxic things, but they're very low levels.
Okay.
Yeah.
How did they take the trans fat out of Crisco?
I don't know. I think that it's not that they take it out. I think they just use a different process to make it in the first place. So they have very low levels of this reaction occurring. Okay. All right. So most of the storage fat, and when a biochemist talks about a fat, it usually refers to a molecule called triacylglycerol. So glycerol is a 3-carbon, this doesn't show up very well, but there are 3 carbons here, and it serves as kind of a scaffold on which to hook these fatty acids. And so if you hide 3 of them, and you use what's called an acyl linkage, then you've got a triacylglycerol. So this is a cartoon, actually an atomic model of the structure. So here's your 3-carbon linker at the top. That's glycerol, which is the same as glycerin. It's kind of a less sweet, sugary type of molecule. And you link 3 of these long carbon and hydrogen chains to it, and that is a fat. And that is a very compact way to store lipids. All right. Now, this, as you can see, it's so compact that there's no water in there. And if you put multiple fat molecules together, the water will not bind them, so they'll tend to bind each other, and you'll get a fat droplet. So in a cell that's specialized for storing lipid, what you will see in this cell is there will be a little bit, there will be the nucleus and the mitochondria and all the organelles will be here, but it will be just one gigantic or just a few fat droplets. And you see these very readily in the microscope because they're so unusual because it's just completely homogeneous, this one type of molecule. Triacylglycerols, and then there may be some other fats in there also. And these cells, anything with a site, C-Y-T-E, that means, like a reticulocyte is a blood cell, a adipocyte is a fat storage cell. And you have many of these cells together, then you get adipose tissue. So the fat storage tissue in animals is just a whole collection. So this one of these cells, if you blew it up, would look like this.
So does the adipocyte have all the other organelles like other cells in the body?
It does. It does. And so that's necessary because its function is to store fat in good times and then to release the fat when it's in caloric need. It needs to get the energy out of it. So it has the whole apparatus to do all the metabolism of the typical cell.
What's the relative size of a fat cell to other cells?
They're a little bit bigger, but not particularly large. And I guess there isn't a size indicator on there, but a micron is a millionth of a meter, and a typical cell might be 10 to 20 microns, and these might be as big as 50 microns. But they're not particularly large cells. So you can see that this adipose tissue is almost pure fat cells too. So, and here I'm going a little beyond what I really know about, but when you think of something like liposuction, etc., the reason they can do that is because you're unlikely to damage other tissues if you just start removing these cells. Okay. All right, so there has to be a whole mechanism to get the lipid into the cells, get them out, etc. We're going to just touch on that briefly in a bit. Now, you see fats, therefore, in this adipose tissue, and it's there as a storage form of energy for hard times. The other major place where you see lipids in the cell is in the membranes of the cell, because that's what really forms the boundary layer of all the cells in our body and keeps the good things in and the bad things out. Because the fact that water won't bind to it means that it's a sort of waterproof barrier. Not completely, as it turns out, but effectively. It prevents soluble things from simply diffusing into the cell. It's a protective coat. And the way it forms this barrier is you have another form of lipid with, instead of 3 of these fat chains, you have 2 of them. And then the third carbon in this glycerol is some kind of molecule that will bind water. So you end up with a molecule with this long part down here that won't bind water and a little part here that will bind water. If you simply take some of these molecules and just mix them together without any enzymes or anything else, they will spontaneously form layers. And what will happen is all of this part will just associate with fatty acids. So you get something like, looks like this. So this is the part that binds water, and it sticks out here where the water is. And then these water-avoiding tails associate with other water-avoiding tails in a layer here, and the ends associate with each other, so you get this bilayer. So in the whole origins of life question, for example, this is one of the most important findings, because the whole question of how do you get some closed compartment in the first place, the answer turns out to be that you don't need any living substance to do that. These compartments will form spontaneously. And if you mix these lipids up and just really vigorously stir them, you'll get little bubbles in there with a bilayer around the outside and an inside and an outside to them. Now, our membranes, if you actually extract and purify them and find out what kind of molecules are in them, it turns out that the lipid is typically less than half the total. What's mostly in our membranes is protein, so that an accurate representation of what the membranes look like is here. So here the red molecules are big proteins that are inserted into this lipid bilayer. And so these proteins form functions like allowing sugar molecules to go from one side of the cell to the other. So your intestine is filled with these transporters. There are receptors that have one end sticking out. They're waiting for insulin out there. Let's say if this were an adipocyte, in its membrane it would have an insulin receptor, and the insulin would signal whether that adipocyte should be forming fats or should be breaking them down and releasing the fat in a useful form into the bloodstream. But the bottom line here is this is why fat is such an important part in making any cell, because it forms one of the key components, which is the membrane around the cell and the membrane around all of those compartments inside, like the nucleus and the mitochondria and all the little vacuoles, all of those structures. A ton of membrane in all of these cells. Okay. And so when you're eating plants or eating organisms, you can get a lot of lipid from the membranes in the cells that you're eating. Okay. No,
Okay, I want to.
let's give it away here. Now, I don't have a sound system. I'm hoping this is going to be loud enough that you hear this. So, when I was teaching biochemistry at one point, I thought I need some way to take this potentially dull subject and put a little life to it. So this is my one and only instructional video, which I always start by saying, if I say that these things form bilayers spontaneously, do you believe it? No, you're scientists. You want to see the data. You want to see some evidence for it. And Rusty has a small cameo role here. You'll see her hand at one point. Okay, so I hope this is loud enough that you can hear this.
Okay, I want to give you a demonstration of how lipids can spontaneously form bilayers in water. So what I have here is some water that has a little bit of ether in it. Now the thing about ether you have to be careful with is because it's highly flammable. And in fact, we're going to test whether in fact a little bit of ether dissolved in this water is indeed really flammable. Okay, I'm going to fill this petri dish right up to the very tippy top. Alright, and if it's really flammable as advertised, we should get a little, whoa, alright, it is flammable. Okay, so, we could put out this flame if we could form a lipid bilayer in the top of that water. So, we need some lipid. Easy source, your ear. Just touch your ear slightly, there, put it right on the surface of the water. What you get is a form of lipid that will form on the surface. It will rush across the surface of the liquid and put out the flame, because there's now a very thin layer of lipid molecules on there. Now, maybe it was a trick. Maybe I put in some retardant or something. Let's give it a test here. It should not stay lit. Little flash, but it won't stay lit because of the lipid layer. All right, now, if it's really a lipid layer there, I have to be able to get a glass rod and pull that lipid layer back, and that's what I'm going to try and do. So I touch it right to the edge, and I pull the lipid layer back, right, get the matches there, and I'm going to have Rusty go ahead and see if in fact, go ahead and light that side and see if in fact the layer is pulled back. Whoa, okay, that really worked. All right, now, if there's really a lipid layer there, I ought to be able to lift up the glass rod. It runs back across the surface of the plate, reforms, and puts out the fire. And so this is a really nice demonstration that in fact just a tiny amount of lipid will spontaneously form an impermeable layer in a water-lipid interface.
Maybe I'll leave this off because it shows up better. Yeah, so it's really just all of the water binding part of the molecule gets its head down, binding to the water, and all the tails are sticking up, and they all associate, and it's just a thin, single molecule thick layer across the top of the water.
What's in our ear? Is that
Well, wax is a form of lipid, so you just have a little. You don't even need to see it. Just the slightest film will dissolve in that liquid and go right across the top.
That's amazing.
Okay.
Mary, is the ether in there necessary to help the dissolving?
Well, the ether there is mostly there for the fire, because because the ether is a solvent for the lipid, but and the trick is keeping it low enough. It's interesting that you can put ether in water and basically turn it on fire, but at the right level to do that. Yeah. Okay.
Barry,
Yes.
what is our skin made up of?
Our skin? Our skin is mostly protein. So one particular kind, collagen for the most part. So the outer layer of the cell, and where that protein is it's outside of the membrane. So you get a cell that secretes through these protein transporters and pores large amount of a protein that will cross-link on the outside. So it's an outer barrier. It's not inside the cell. Okay. And just a little aside there, can't resist. Have you ever wondered why these bloodhounds can follow a trail of a person 2 days after he's gone? You know, even if that person was pretty smelly, the wind would have come along and blown that away. It's a direct answer to your question, and that is that as I walk across this room, I am leaving a trail behind me of skin cells. And it's my collagen and all of the proteins, etc., and it falls onto the floor and will persist for days. And dogs have so many scent receptors that they can go along the ground and pick up that particular pattern of collagen and skin proteins and follow it behind people. So anyway, wonders of biology. All right, so you've heard that in your diet, there, you want the light back on? Okay.
We need it for the film.
Oh, that's true. there are certain essential fatty acids. So in the very first day, we talked about things that you must get in your diet. You can make most of those fatty acid chains, but you can't make everything. And the types of fatty acids that you cannot make are those that have double bonds towards the end of the fatty acid chain. So there's that acid group on one end, and this other end is called the omega end. So you've heard about omega-3, omega-6 fatty acids. They're required in your diet. So here's number 1, 2, 3, double bond. That's an omega-3 fatty acid. I was looking at this just before the class, and this linolenic is also required in our diet. It's an omega-6. And this diagram that I took from I don't know where now is wrong, because it's got it at 1, 2, 3, 4, 5, 6, 7. And I looked it up, and it should be at number 6. So omega-6, double bond, 6 from the end. Omega-3, double bond, 3 from the end. These particular kinds of fatty acids, which we do not have the enzyme to put the double bond in that particular position, these amino acids form this kind of unusual structure, and then it's sort of like ring-like, because these kinks towards the end cause it to twist around. All right? And this kind of summarizes this sort of dietary situation here. So the omega-6 types, linoleic acid is called, and you can get them from plants, soybeans, corn, corn flour, corn, canola oil, etc. All right, and this is not the actual final product you need, but your body has the enzymes to convert this to more complicated structures. And this final one here called arachidonic acid, and you can get that out of eating meat, because other animals have to make these. So the cow has eaten the plants to get the precursor to make arachidonic acid. So you can either eat the plant and convert it to yourself, or you can eat the cow who has already made it for you. These are involved in inflammation. So, for example, the pathway that aspirin affects, anti-pain or pro-pain inflammatory responses, are signaled by molecules like arachidonic acid. Now, the omega-3s here, which are present in a smaller range of organisms, so they mention here like flax, hemp, chia seeds, etc. They are converted to something that is most abundant in certain kinds of fish, oily fish. So they often say fish are a good source of these omega-3 fatty acids. All right, and so you can get this to make other things which are also signaling molecules operating primarily in the inflammatory response. So the number of steps for this conversion is a lot of steps. It's somewhat complicated biochemistry. And basically, if you have a purely vegetarian diet, you can get enough of these in the end, but your body does have to convert them. And it's not very efficient in doing that. And that's why they often recommend that you have some fish in your diet, because this step is something your body can do, but it doesn't do it very efficiently, and you can end up with deficiencies if you're using just the plants. Plus, these are not, your kind of typical plants. We don't eat a lot of chia and flaxseed in places outside of Santa Cruz. Other places, that's not the case. Okay, so what we do with fats, for the most part, far beyond our need for making membranes, etc., is we use them for energy. So fats are high-calorie foods, which is a way of saying if you burn them, they generate a lot of heat. If you eat them, they can generate a lot of energy. They can generate a lot of ATPs that we talked about last time. So you have a single fatty acid chain from one fat molecule, and that can generate 108 ATP molecules. So they're really high energy compounds. the way it's done is pretty straightforward fashion. There are enzymes in your cells that will chop off these carbons 2 at a time. They'll be cut in pairs. They'll take off 2 here. That'll make what's called an acetyl group, so 2-carbon molecule. That'll go into the whole ATP producing pathway. And there's really fascinating enzymes that do this. They kind of bind the fatty acid at one end, and then it clips off a piece, and this actually requires several reactions. So it's kind of a cycle. It goes through one cycle, cuts off 2, goes through another cycle, cuts off another 2. It just keeps chopping them off until you're down to the last 2 carbons.
Is ATP synthase part of this mechanism?
And I'll show that next. Yes. And the fact that they're cut into is why fatty acids are typically in 2-carbon increments, so you'll have 16, 18, 20 carbon fatty acids. It's also because that's the way they're made. They're made out of these 2-carbon units. So when the cell is synthesizing them, they put them together in even numbers. All right. So to address your question, we looked at this metabolic chart before where this is glucose and it goes through where the 6-carbon glucose gets split into a pair of 3-carbon molecules here. And then in the very next step to glucose, you produce a 2-carbon acetyl-CoA, an acetyl group, 2-carbon. When you degrade fats, it enters the pathway right here. And then these acetyl groups go in and go around that cycle of reactions where the 2 carbons end up as CO2, and the electrons in the molecule all end up on the NADH, and it goes into the same ATP synthesizing system that we talked about last time. Oh, and let me just mention, as far as last time, we didn't video the last one, so I did it at home later, and I tell you, it's so much harder to do without an audience. So the content is available there, but it just doesn't have much oomph to it. All right, okay. So the other major important type of lipid in our diet are the sterols. So what makes something a sterol? It's this ring structure here. So this looks very different from that long fatty acid chain, but what is somewhat in common is there are not very many oxygens involved. So all of these rings here have no oxygen except for one right here at the end. So in this whole molecule, where on it does it have a sort of slight positive or negative charge that could bind a water molecule? And the answer is only here, only in this OH on the end of the molecule. The rest is going to be hydrophobic, not water binding. And the structure of cholesterol is shown here. Plants have similar sterols. They just have minor variations in these little side chains here that make them something like ergosterol or brassinosterol, etc. They're all fundamentally very similar. So what it really looks like, in this case, the kind of illustrative chemical structure doesn't really show you very well what the molecule actually looks like, because it really looks like this. And when you look at this, it's not so different from those lipids that are in the membrane, where you have the cholesterol and 2 chains. It's basically a lump of hydrocarbon. And that's the same thing here. You have a water-binding part and a just big hydrocarbon, kind of lumpy chain, if you will. Okay. Now, it presents a problem in our diet because it's not water soluble. And so how does it circulate in our digestive system? How does it circulate in our blood? And this problem is at the core of a lot of cardiovascular disease. Now, obviously we have evolved ways to deal with it, but it still presents a dietary challenge to our body. On the one hand, you've got this, you just ate a big pat of butter because it tastes good, and now you've got this lump of stuff, and your body wants to get it inside because it knows it's just a super source of ATP, and you never know when you might need more ATP, so let's get all we can get. But how do you get it across the membrane? How do you get it flowing in your blood, etc.? So there's a whole elaborate transport system that has evolved to deal with that. So here's the inside of your intestine, and what will happen is there are carriers in the membrane here that will take it into this intestinal cell. Now, once in the cell, it will be complexed with other types of proteins, and in some cases, it's going to be packaged in a little membrane vesicle of its own, and that vesicle will fuse with the cell's own membrane, all right, and then be released out here. Some of it can go into the blood, some of it can go into the lymphatic system. Okay. And this is where HDL and LDL come up. So, we've all seen this when you do your blood test. They tell you that it's important to, when they talk about keeping your cholesterol at the right level, they don't actually measure cholesterol per se. They measure the carrier molecules that contain cholesterol. And there are about half a dozen major types, but they usually focus on these two, HDL and LDL. So, what this stands for is high density adipocyte, etc. And so that's the function of the proteins, to actually form an address, to participate in the transport mechanism. They also probably form a nucleating structure to initiate the formation of these droplets. Now, is lipid a relatively high density or low density molecule? Well, when you pour oil into a pan, does it sink or does it float? And it floats. It's low density. It's less dense than water. And I hope I don't get into trouble here, but do women float better or men? Women float better, probably because of pregnancy, the potential for childbirth, etc. Women have a thicker fat layer in their skin than men do. All right. So something we all knew before: men are more dense than women. Nothing we can do about it. It's just our biology. Okay. Now, LDL is the same basic structure, but it has fewer protein components. So the higher ratio of lipid to protein in LDL, and that's why it's low density, enriched in fat, versus HDL, which is high in fat. Now, also a great simplification, but you can see that there are not a lot of helper molecules here to keep this from associating with other lipid molecules. This is more likely to form some sort of plaque, clump of lipid in your veins and arteries than the HDL. And that at a very simple level is why this is considered the good cholesterol and this is the bad one. And it's the ratio of these two that's the best indicator of, just from epidemiology, whether it's associated with cardiovascular problems. So, so exactly what underlies this sort of plaque formation, etc., is not simple, and it's not just a solubility issue. And they have to have sites at which these plaques initiate, and there are probably other defects in the whole transport mechanism. But in a very crude level, basically LDL seems to be involved in forming these plaques, or if you've got too much of this, more than the blood can safely transport and kind of dissolve, if you will. then it can be counteracted by the HDL, which is more efficient in solubilizing it and carrying it to other parts of the body. All right. Questions on that, HDL, LDL, cholesterol?
So even if you eat low saturated fats, your liver is still producing cholesterol.
That is correct. Yes.
So you could have high numbers, just as long as you have more HDL, then your ratio is good.
Yes, possibly so. And you find this, that diet may or may not be a cause, and it all depends on your kind of personal physiology. So some people can control their cholesterol by controlling cholesterol uptake in their diet. For other people, no matter how careful they are about that, they're just going to have high cholesterol. And it's usually due to some variance in a gene that encodes one of the proteins that is involved in making these lipoproteins, or in the transporters that interact with them. I'm going to address a little bit more of that in a bit too.
Where's the HDL taken? You said it carries it off to other parts of the body. And it's sort of a cow.
Well,
where's it going?
Can go to almost any cell, but the major destination from your intestine typically goes to your liver, and your liver then does the package and sending it. If it's in excess, it will go to an adipocyte, and it'll just be stored there. Another place it can go to in sizable quantities is your muscle, because your muscle has major fat storage in it. And you think when you're eating cows, the best ones are those that are just loaded with fat in the muscle, and that you're just looking at the biology of lipid storage in that muscle tissue. But it can go to other kinds of tissues as well. Doesn't go to your brain. Your brain will only take glucose. Don't give it any of that fat stuff. It just wants sugar, pure sugar.
What do statins do then?
We're going to come to that next.
Okay.
Yeah.
How does the lipid attach with the blood vessel?
I'm not sure, I don't know the answer to that. And it's one of the complexities of understanding how this gets initiated to begin with. Because blood vessel is made up of cells, so it is a question, is it just physiology, is there just an excess, or is there something different in the chemistry of those spots? I don't know the answer. I know that's one thing that's really intensively investigated, and it's complicated. It's a very poor answer. Okay. So, statins. All right. I would bet that probably nearly half the people in this room probably take statins. So what are statins? Okay. And this brings in, it's a drug. All right. And this raises the question I love to ask people, what are drugs? Chemically, what's the function of a drug? Why do drugs work? Now that you're partly trained biochemist, what would you say?
They target a change. They target and change.
So what do they target?
Whatever they're trained to target.
Ah, trained to target. What do they target?
Receptors.
So that's a good answer. A protein, type of protein, receptors. And what are most proteins? Enzymes. Most drugs are inhibitors of enzymes. All right. So if you take aspirin, you inhibit the enzyme involved in that arachidonic acid pathway, which is a signal for inflammation. So if you inhibit the enzyme that's making the inflammation signal, there's no inflammation, and that stops the pain. So if you kind of go through the whole list of different kinds of drugs, I think of for some reason, different kinds of drugs that you can take. In almost every case, you are targeting the binding site of some particular protein, and you're trying to stop some metabolic reaction.
What do opiates do?
What opiates do is they go to receptors, which are not really triggering chemical reactions per se, But what opiates do is bind to a protein that affects whether ions move across nerve membranes, and also whether other signaling molecules that turn on and off the electrical currents in neurons. And so they'll either turn them on or turn them off in certain neurons. So that's how they affect your nervous system. So they can affect kind of pleasure responses by turning, say, on the enzyme that secretes certain, dopamine and related compounds like that then go on and trigger a whole bunch of neurons. But all of these drugs are basically chemical inhibitors of enzymes. And statin is a classic example. So HMG-CoA reductase is an enzyme that uses acetyl groups, that's the A, puts them together in a form that makes cholesterol. So it's the key step in the synthesis of cholesterol. All right. If you had a chemical that looked just like this, but would bind to that enzyme and not come off, you would inactivate the enzyme that makes cholesterol. And that's what statins do. So there are many different kinds, lovastatin, simvastatin, etc. And if you look here, this doesn't form quite a complete ring. These have almost exactly the same structure with just some slight modification. The consequence is they fit very tightly into the pocket of this cholesterol-making enzyme, but they won't come, they'll go on, but they won't come off. All right. And so the reason they're so effective is that they can really be given at the right dose and lower the production of cholesterol by your liver. All right. So this is one of the studies of statins, and what people were studying here was the treatment of this disease, familial hypercholesterolemia, sort of relating to your question. So familial means it's an inherited disease. It's a genetic problem. High cholesterol in the blood. So this is a fairly common genetic mutation, and people who have the most severe forms, I don't know what good cholesterol numbers are, like 120 or something like that. These can have cholesterol numbers of 500, even if they're pure vegetarians, and that's simply because their liver is overproducing cholesterol. And it's found that if you treat them with statins, if there's no statin treatment and you're looking at cumulative event-free survival, in other words, are they having strokes or heart attacks? If there's no statin treatment, survival goes down. Just over a 10-year period of this study, the quite dramatic drop in survival. People are dying of strokes and heart attacks with this disease. Whereas with the statins, almost no effect. Really aligned very much like that of the general population. Probably one of the most prescribed and one of the most successful drugs in the last 20 years. Very few side effects. In fact, there's a long-running controversy. Men tend to take it more than women. There's a question of whether women should be taking it. Some people advocate everybody should be taking it. But it does really seem to work without many side effects. There are always some, but relative to most drugs, it's a very good one.
So you reminded me of last week, we were talking about antioxidants.
Mm-hmm.
And taking antioxidants, your opinion, I think, is that don't bother,
As a supplement, right.
because your body produces what it needs and takes care of itself. And now we're talking about statins, and we're saying, no, you need to have this. But your body's producing what it needs, and you overdo statins.
Yeah, and so it comes down to a question of whether you have some sort of mutation family history that indicates that your body is an overproducer. Most of us are not. So for most of us, the problem is we make plenty, we make about the right amount. But then if we get an additional dose in our diet, then we end up with high levels of cholesterol.
Is the additional dose in your diet a temporary thing? I ate a lot
Well, no,
this meal or something?
so I think the point is that if, for most people, simply paying attention to their diet and keeping total cholesterol uptake, which is basically meat uptake, at a kind of low level, their cholesterol will remain at the right level. It's just, so it's diet induced. But there are quite a few people, and I would be guessing, but maybe as many as 10% of people who have some genetic mutation that they just kind of naturally produce more cholesterol. And the diet will often help. People with the worst forms, diet won't help at all. They just have an internal overproduction problem, and that has to be dealt with by some kind of drug.
Yeah, it's probably worth adding that there are alternatives to statins that are very, very effective with zero side effects. In Ayurveda, Google is used, and so far it has proven to be equally as successful with no side effects.
What's it called?
Google, G-U-G-G-U-L, it's a natural product.
Okay.
And in Chinese medicine, they treat the liver. They say that the liver needs to be calmed down. And then oftentimes they've had success with that and then been able to lower the statin or even eliminate it entirely.
Okay.
Barry, you mentioned that the brain loves sugar and
Right.
and doesn't really absorb fat or doesn't.
Right.
So how does a statin impact the brain?
It probably doesn't have any effect, but that's okay because its target is the cells that's making cholesterol, and there are not cholesterol-making cells in the brain. I shouldn't say that as a blanket, but effectively no. It's primarily going to the liver because that is the primary site of cholesterol synthesis.
In your previous slide, you showed that lovastatin and simvastatin were colored in green, and all the other statins were. Is that because the chain is broken on the other statins, or what is the?
I hadn't paid attention to that. I don't know the answer, so I better not guess.
Okay, it's okay.
Okay. There were other questions. Yeah.
I just want to, you keep saying that targeting lowering cholesterol. Is it only lowering LDL?
Well, what in this case, what it's really lowering is the cholesterol itself. LDL is the carrier of it, but it's targeting the cholesterol. So that gets into the complexity of that. It's not that simple. So it depends if you have a genetic predisposition to high cholesterol. It could be that it's not just the level of cholesterol being made, but it could be in the proteins that make the different carriers. And let's say you had a defect in the HDL carrier. So it may be that you knock your cholesterol down, but you still don't have the right level of the beneficial carrier. So there are all of these complexities out there also.
So back to ratios,
Mm-hmm.
I'm from that, I have that genetic disposition.
Yeah.
And that's what caused me to go to a plant-based diet.
Mm-hmm.
I was able to get my cholesterol down to the low 200s, but my ratio is excellent. Can you explain why that is fine now?
No. Yeah. I just don't know enough about that to say.
Okay.
Yeah. And I suspect that a really good answer is not totally available yet.
Okay.
Yeah. Okay. So we talked just a little bit about proteins, our last major class. How are we doing on time here? All right, so protein-rich foods, and there are not very many foods that are really pure protein. Chicken is pretty good. Actually, chicken is relatively low fat, etc. Some kinds of fish. All right, but like this steak in the middle is they're showing a very fatty steak here for their picture on a protein-rich food. All right, So proteins are made out of amino acids. The basic structure is this, an amino group. This is an acid group. This simple 3-atom core to it is in all amino acids. And then the R group is some side chain there, which can vary from something simple like a hydrogen to a little fatty acid chain, if you will. And I'll show you that there are these 20 different kinds, really hard to read. But the point is that in some cases, this R group is just hydrogen. This R group is a CH3, a methyl group, no oxygen there. This R group has 3 of these methyls, no oxygen. The fact that there's no oxygen in these means that they don't bind water very well. So these are sort of fatty-like amino acids. They're going to associate with lipids. Then there are others that have side groups, OH, nitrogens here, that do develop a charge. They will bind water. And there are others that are kind of in between. So you have these 20 different kinds that make up all your proteins and all your enzymes.
Why don't we turn out the lights for this?
Yeah, for this one. Yeah. Okay.
Yeah.
So, in our bodies, we have lost the metabolic pathways for about 10 of the amino acids. And we get them from our diet. Every creature has these 20 amino acids, animal, plant. And so we don't really need them. We don't really need to make them if we're eating other creatures. But this is going to be true if we have a reasonably balanced diet. But there are ways that problems can occur. We touched on this briefly at the beginning. And that is, so if we look at, if you're a real vegetarian, it's important to know that you shouldn't specialize in any one particular kind of plant, because different food sources have different limitations in the amino acids. So what I mean is, if you eat beans, they don't have very much methionine in them, and that's an essential amino acid. In fact, vegetables in general don't have very much methionine. So where can you get them? Well, they're stored in grains in fairly high quantities. Grains, on the other hand, don't have much lysine or threonine, all right? And nuts and seeds are lacking lysine. So what this table shows you, that if you eat beans, you're missing methionine. However, if you eat beans and rice, then you're going to get the methionine from the rice, you're going to get the other things from the beans, and you're going to be okay. And corn is an interesting one. If you have a very high corn diet, you're going to be missing lysine, you're going to be missing this amino acid tryptophan. And so how do you supplement that? Well, a good thing is to eat some beans and peas, etc. So this is really an old, well-known fact in diets, but it's a problem to this day. So there are many parts of Africa, for example, where people eat corn breakfast, lunch, and dinner. They essentially eating something like polenta or grits. It's basically cornmeal. And if they eat just that, they will and do, unfortunately high levels, develop severe nutritional deficiencies, and part of it is a lack of some of the essential amino acids. But if you eat a mix of the things, you'll get all of this. It's because plants, although they have the same total things, have a different ratio of the amino acids than most animals do. Okay, so kind of make some kind of overall comments here at the end. First of all, let's look at this important bit of biochemistry. So ounce for ounce, fat is high calories. So 1 ounce of pure fat will have 250 calories. 1 ounce of pure sugar, about 111. 1 ounce of pure protein, about 92. So these two are very similar, but fat is about 2.5 times more caloric than the others. It means it has more energy And why is that so? So, you're all becoming almost fully trained biochemists here. Here's what we've got in protein. Here's what we've got in the sugars. And here's what we've got in the fat. What's the big difference between the fat and the other two? Which? Well, more carbons means less, fewer what? Oxygen. Oxygen is the key. So this has oxygen in it. It also has this amino group that has less energy in it. Sugars is loaded with oxygen, but this carbohydrate chain has none. How do we get energy out of our food? What's the last step in the pathway? We combine those electrons with oxygen. These are already, a lot of these electrons are combined with oxygen. So it is literally partially burned food. We combust, burn our food, we combine it with oxygen, and these molecules allow us to pass fewer of the electrons onto oxygen to make ATP because the oxygen is already there. This, on the other hand, is just pure electrons, all of which can go into that ATP synthesizing pathway. And one day I was listening to NPR and they said, they were talking about the controversy over growing corn to make fuel for your car. And the reporter said, did you realize that an SUV, that the corn you need to produce the ethanol to fill up your car one time will feed a person for a year? And I said, that can't be right. So I went back to my office and I did a little scribbling. And he's right, absolutely right. And a part of the problem is that ethanol is this is what you're putting in your car. You're converting cornstarch into ethanol. It's a fairly low energy molecule compared to gasoline, which is just pure hydrocarbon. So you look at this structure of gasoline, what does it look like? It looks like those lipid chains. It's exactly the same molecule. It doesn't have the acid group at the end. But the hydrocarbon chains in a Hershey's chocolate bar are the same as the hydrocarbon chains you put into your gas tank, all right? And the energy equivalent is the same. Methane is also has no oxygens in it, so it's high energy. So by the time you grow the ethanol, and then it takes 30% of the energy in the ethanol to separate it from the water molecules that were produced in its fermentation, you really have a very inefficient use of the energy that was in those corn grains to begin with. Okay, so final things to say. General thoughts about diet. Let me just give you 2 slides here. One, you see this? If you order it, make sure it's going to be shared between 3 of you. Okay. This is a real sign. Took it, Rusty, I said, we're driving down the road in Mobile, Alabama, and I said, Rusty, you've got to get a picture of that IHOP sign there. What's the major problem with the American diet doesn't have anything to do with chemistry. It's just that you can get so many calories for so little money. 2 pork chops, 3 eggs, and 3 pancakes. This is probably 3,000 calories. It gets through a day and a half just by itself. So my summary of kind of nutritional advice is, first of all, the calories do count. I mean, if you're taking in a lot of calories, your adipocytes are going to have a great time storing that fat. All right, you will keep them. And the calories are often coming in the form of fat, and they're stored in the form of fat. Now, having said this, there has been quite a bit of rethinking about whether is fat in your diet bad for you. And right now, the sort of consensus is not if you aren't taking too much. So it's more a question of total calories than whether fat per se is an evil molecule. As long as it's not a trans fat, etc. As long as you're just not eating really high cholesterol food, you can deal with it. All right, and then the bottom line here is really that balance is the key. You know, you want to eat a bit of everything. And one nice recommendation I think that has come out of these national nutritional advisories is that the easiest way to see if you're eating the right thing is to look on your plate and see, is it all brown? If it's all brown, it's not a really nutritious meal. If it looks like this and it's all brown, it's a severe deficiency there. So if it looks like this and you got some green, you got some red, you got some orange. So here you've got mostly plants, you've got some carbohydrate here, and you've got protein there. What's missing here is the whole vegetable component. Perfectly good food, but it's deficient in many things. And this is my kind of second to last point. It's best to eat cells. You can see these are cells. This is processed. All right, this is cells. Here, there's almost nothing here that's cell. Well, the hamburger is. Okay. And why? Because every one of those cells has about 20,000 compounds in just the same mix that's in your body. So it's a good balanced meal, almost by definition. And lastly, there are no magic foods. They appear every 6 months. Be skeptical, all right, all right. And, you know, this is probably the key take-home lesson. All right, so I want to thank you all for participating. All right, great audience. And we'll put this one on the video too. I'll probably do this again next year as long as I have customers. All right. And you might, if you just have a few minutes, you might just walk up one floor and look around this building. It's a really interesting building, the layout of it. Basically, in the center here are all the offices. So the undergraduate students can come in here and find the faculty in their offices. But then in the wings is where all the laboratories are, and the graduate students and the postdocs, etc. And as you go around, they have the names of a lot of famous scientists, Darwin and Mendel, etc. And one day I was walking down these stairs, and there was a student who was showing another student around. And the one student said to the other, so these names here are these all the faculty here? Oh yeah, Charlie Darwin. He's got an office right down there. All right, so thank you very much. Okay.
Thank you.